This is Unit 3, Cells, Part C, Membrane Systems. The first part, it deals with membrane location and composition, transport through membranes, selectively permeability, and characteristics of transport proteins. Because Part C, Membrane Systems, is rather complex, I've divided it into 3 separate videos. As we mentioned, this video covers these 5 topics. Part 2 will discover concentration of solutions, concentration gradients, passive and active transport. And Part 3 deals with cell-to-cell communication and other membrane functions. Cellular membranes are located around the cell in the form of the plasma membrane and inside the cell, associated with the membrane-bounded organelles. Membranes are composed of lipids, proteins, and carbohydrates. The lipids include phospholipids. They make up roughly 75% of the membrane lipids. They have a variety of functions. They form the basic sheet of the membrane. It's called a phospholipid bilayer. They modulate protein activity, and they generate second messengers. We'll have more to say about this at a later time. Steroids make up about 20% of the membrane lipids. They are found within the membrane, and they serve mainly to provide strength to the membrane. They consist mainly of cholesterol. Another type of lipid are the glycolipids. Roughly 5% of the membrane lipids are glycolipids. They tend to form an outer surface of the membrane. Proteins make up 1 to 10% of the number of molecules, but because they're large, they make up approximately 50% of the weight of a membrane. Integral proteins are those that are firmly attached, partly embedded in the membrane or extending all the way through the membrane. Peripheral proteins are loosely attached to the surface of the membrane. The proteins have a variety of functions. They may act as transport proteins. We might call them channels or carriers. They act as receptors. They receive signal molecules. They serve as enzymes to catalyze reactions. Many of them are used to link one membrane or structure to another. We could call them adhesion proteins or linkers. They serve as motors to move structures around within a cell. Many serve as identity markers. We could call them recognition proteins. The complexity is illustrated by the fact that many perform more than one of the above functions. I would say it's safe to say that any membrane protein performs 3 or 4 more functions. Carbohydrates include glycoproteins and glycolipids. They form an outer covering called the glycocalyx. Transport of particles is mainly through membranes. This includes diffusion, osmosis, dialysis, facilitated diffusion, active transport, endocytosis, and exocytosis. 2 categories of transport are passive processes that do not require metabolic energy and active processes that do require metabolic energy. The net movement is determined by the need for energy. Net movement from high to low concentration does not require metabolic energy. You'll see more about this later. Transport, though, from low to high concentration does require metabolic energy. Based on the involvement of a transport protein, transport may be carrier-mediated, which does require a transport protein, or non-mediated, which does not require a transport protein. We'll come back to these examples after a little background. Carrier-mediated transport includes facilitated diffusion, active transport, and some forms of endocytosis. Non-mediated transport includes osmosis, some dialysis, and some filtration. Membranes are selectively permeable. That is, some particles will cross and others do not cross. The following cross through the lipid bilayer, usually forms of non-mediated transport. They include lipids, because after all, lipids easily dissolve in the lipid bilayer. Remember, likes dissolve likes. Non-polar molecules, such as carbon dioxide, oxygen, nitrogen, and small polar molecules, even though polar generally don't mix with nonpolar substances, they're small enough to fit through the gaps between the phospholipid molecules. Urea is an example, ethanol another example, and water. Now this is a slow form of transport, and even though water does not mix with lipids, the water molecules are small enough to work their way through the lipid bilayer. Now, water also crosses through membrane proteins. These are called aquaporins. Following cross with transport proteins, mostly forms of carrier-mediated transport. Large polar molecules, because they cannot fit between the gaps in the phospholipid bilayer. This includes glucose, amino acids, and many, many others. Ions absolutely can't cross the phospholipid bilayer, so they require transport proteins. Sodium, calcium, fluorine, and potassium, and many other ions as well. Transport proteins come in several types. Some are holes. They may be channel proteins, ion channels, or other channels. Some are carriers, pumps, receptors. I may use these terms interchangeably, and they generally refer to the same functional protein. Transport proteins are gated, controlled. That is, they can be opened and closed like a gate. Voltage gated or opened and closed by ions. Ligand gated or opened and closed by molecules. Mechanogated are opened and closed by direct contact. Let's look examples of gated transport proteins. Voltage-gated example would be ion channels in nerve cell membranes that, when opened, allow potassium to enter the cell. An example of a ligand-gated protein, molecules in the nose that are sensed as odor would allow the sodium to enter the olfactory cell membrane, leading to the sense of smell. Mechanogated include pressure on the skin, for example, which allows sodium to enter a pressure sensor. In the ear, sound waves actually tug on a protein cord, opening a lid, allowing potassium to enter and leading to the sense of hearing. Transport proteins work by binding to the molecule being transported, flipping to the other side of the membrane, and releasing the molecule as shown below. Three characteristics of transport proteins are specificity, competition, and saturation. Let's look at specificity first. Assume the square molecule is to be transported into a cell. Here you have 3 cells shown. Each cell has a transport protein in its membrane. Only one transport protein is complementary to the square molecule. Which cell possesses a transport protein specific to the square molecule? Cell number 3. Competition. A different molecule with a similar shape competes for the same transport protein. Different molecules can bind to the transport protein and prevent the square molecule, for example, from binding. An example is antihistamine. Saturation. Saturation means filled to capacity. Here is a cell with 3 transport proteins. Their role is to transport molecules across the membrane into the cell. In the extracellular fluids are different concentrations of the molecule to be transported. Assume a transport protein can transport 1 molecule per second across the membrane. If the concentration of molecules in the extracellular fluid is 1, how many can be transported per second? 1. If you raise the concentration of molecules in the extracellular fluid to 2, how many can be transported per second? 2, because they're both operating in that second. If the concentration of molecules is increased to 3, how many molecules can be transported per second? 3, because again, they're all operating in that 1-second interval. If the concentration of molecules in the extracellular fluid is 4, how many can be transported per second? The answer is 3, because there are only 3 receptor molecules. If the concentration increases, the number transported remains at 3 per second. And this is the end of the first part of Part C, membrane systems. You're now ready to continue with the second part.